Hello, welcome to our live session for tonight. I'm going to be reviewing some things that you need to know for the Chapter 5 and Chapter 7 test. So just as a reminder, Chapter 5 is about enzymes, and Chapter 7 is, it has a long title. It's like how cells make energy or something like that. But I think that's the title, how cells make energy. But the actual topic is aerobic respiration and fermentation. And I encourage you to watch my video on aerobic respiration and fermentation. It takes the detailed information from the book and it identifies really what you need to know for the Chapter 5 and 7 test. I've mentioned it in the instructions for for week 6 that the book for Chapter 7 expects you to know way more than what the test will ask you. So watch the video. I encourage you to do so. So we'll start with Chapter 5. What is it that you need to know? So the big ideas when it comes to enzymes is, number one is, what is an enzyme? That's one big idea. And I'm hoping, let me adjust my light here. I'm hoping that with the information, you know that an enzyme is a protein molecule that regulates chemical reactions. I'm going to say it regulates and speeds up chemical reactions. And again, the video talks about, this business about speeding up and regulating chemical reactions. You should know some characteristics or features of an enzyme. I don't remember what I called them in the video. Did I call them characteristics or did I call them features? So some things to remember is that enzymes are catalysts. And catalyst is anything that speeds up a chemical reaction. Because enzymes are protein, sometimes they're called living catalysts. Or sometimes they're called biological catalysts. Either is okay. I promise you will see the word catalyst on the quiz. So remember that catalyst is kind of interchangeable with enzyme. Some other features that you'll need to know about enzymes is, what is the business? What is the relationship between a substrate, an end product, and the active site? You'll also see on the test the word enzyme substrate complex. So you need to know what these words refer to when it comes to an enzyme. Oh, another word here is turnover number. I think in the quiz, they call it the turnover rate. Okay, so the turnover number or the turnover rate is the same thing. You need to know how enzymes are affected by a change in temperature and a change in pH. And I will tell you that a word you need to know associated with those changes is what does it mean to denature an enzyme? How does that affect the turnover rate? Some other things that impact your enzyme is the amount of substrate that's available. Just as a reminder, if you increase the amount of substrate, generally speaking, your enzyme works faster. There's a limit, of course. You can overwhelm your enzyme, but if you want it to work a little faster, you can give it more work to do. And so if the substrate goes up, then the enzyme works faster. The converse is also true. You can slow down your enzyme by lowering the amount of substrate. That also slows down your enzyme, or it slows down the turnover. So let me label these A, B, C. You can also make an enzyme work faster by adding an enzyme helper in the form of a vitamin or a mineral. Minerals are usually metals such as you can have iron. Iron can be helpful to make some enzymes work better. And vitamins such as vitamin B, vitamin C, they are also, if you add them to an enzyme, they make the enzyme work better. So generally speaking, if you add these, you increase the turnover number. So there'll be some questions about how an enzyme, how you can make it work better, or how you can actually make it slow down, or in some cases, how you can make it stop working. Let me think here. What is another question about enzymes? Let me double check real quick. I'm going to pause the video so I can check one thing. Okay, so I just checked the chapter 5 and 7 test to make sure that I covered, or I listed everything that I know can appear about enzymes on the test. So let me talk now about chapter 7. What are the big ideas from chapter 7 that you might see on the test? So chapter 7, and the title, I looked it up while I was away. How cells make energy from food. And again, that's the title of it. It's a really long title, but the two topics are aerobic respiration and fermentation. So here are the big ideas. Things that you need to know that you'll see on the test. One of the big ideas that you'll see is, what is ATP? And I hope that you know that ATP stands for adenosine triphosphate. And it's the energy molecule of all cells. This is an important topic in biology. If you take 100 biology classes, you're going to hear about ATP and its importance. So ATP is like a universal thing that we talk about in every biology class. It's that fundamental of an idea. It's that important to every biology class. And so just as a reminder, ATP is made up of an adenosine part, and then triphosphate. Triphosphate means that there's 3 phosphates attached to that in adenosine, and the last 2 phosphates are usually connected by a wiggly line to indicate that there's a lot of energy stored right here. There's energy stored in every chemical bond, but this is the bond that gets broken and reformed most easily. So we pay a lot of attention to that particular bond. So if you break that bond, then you release energy, and the cell is able to use that energy for whatever work it needs to do. So it could be to move something from one side of the cell to another. It could be to take food in or take waste out. Whatever the cell needs to do, it needs energy to do that thing, and it gets the energy by breaking that chemical bond. That's why ATP is so important. But chemical bonds, they contain energy. So when we break them, we get the energy out. So how does that relate to aerobic respiration and fermentation? Well, the big idea is that, and I'm going to abbreviate, aerobic respiration and fermentation are two ways cells can generate energy. Okay, that's a big idea. There are two ways that a cell can generate energy. One way, aerobic respiration, it requires oxygen in the form of oxygen gas. You'll see oxygen gas abbreviated like this a lot. Whereas fermentation, it can happen in the absence of oxygens. That's why sometimes it's called anaerobic respiration. Now, both aerobic respiration and fermentation, they're basically a series of chemical reactions. That's what they are, is a series of chemical reactions. You're not required to know the chemical reactions. I'm not going to expect you to diagram those reactions or write them out in any way. There's some generic diagrams that we show in the chapter 7 video, but we're not going to go to the detail that your book is showing. I'm not discouraging you from reading your book. I'm just telling you that your book is going to explain more detail than I would expect you to know for the test. Here's what you do need to know about aerobic respiration and fermentation. You need to know where in the cell does that process occur. You need to know what do you need to start the process? So what are the starting materials? What are the ending materials or the ending products? You need to know, does that process require oxygen? So again, I'm going to advertise for my live session from last night. Go back and watch the video on aerobic respiration fermentation, because I do some diagramming a little bit, and I answer these questions real specifically. Where do those processes occur? What is the starting material? What is the ending products? And does that process require oxygen? Here's a neat little diagram that kind of shows the relationship between aerobic respiration and fermentation. So there's a process called glycolysis. And glycolysis is like the beginning of breaking down a sugar molecule. What happens after glycolysis is dependent if you have oxygen readily available. If you have oxygen readily available, glycolysis is going to be followed by the Krebs cycle, and then the Krebs cycle will be followed by what's called the electron transport system. I believe your book calls it the electron transport chain. So I'm old school, I call it the system, but you can also call it the electron transport chain. It means the same thing. So when we refer to aerobic respiration, really and truly, aerobic respiration is the last two parts. This is really aerobic respiration. Your book includes glycolysis as being part of aerobic respiration. And really and truly, it's both, it's just these two parts. It's not bad if you include glycolysis, but what is confusing is that glycolysis is an anaerobic process. But everything that follows glycolysis requires oxygen. What happens after glycolysis if you don't have any oxygen or if it's very limited, then the breakdown of sugar continues through fermentation. And there's two types of fermentation. There's what we call lactic acid fermentation, because you produce lactic acid. And then the other type of fermentation is called alcoholic fermentation, because you produce, you guessed it, alcohol, specifically ethanol, the kind that you drink. So one thing that you should know is that muscle cells, like that's inside of you, muscle cells from animals, they undergo lactic acid fermentation. And yeast and some types of bacteria undergo alcoholic fermentation. Two more on this list, some big ideas, and that's going to be the structure of a mitochondrion. What does it look like? By the way, mitochondrion is singular if you're talking about just one. If you use the plural word, it's mitochondria. The ending's a little different. So for this mitochondrion, what does it look like? How's it built? And so when you're answering the question, where in the cell do these processes occur, you'll relate the location of where these processes occur to how the mitochondrion is built. During the breakdown of sugar, I talk about these things called electron carriers. There's two of them. There's NADH and FADH2. And so I would know what is an electron carrier and why are they important to aerobic respiration. They're actually important in fermentation too, so we'll just say, why are they important? Did I say I only had two more things? I meant three more things. The last one is, how many ATP on average are produced from the breakdown of one sugar molecule? And really not just any sugar, specifically from one glucose. So if you're wondering what you need to know for the chapter 5 and 7 test, there you go. Those are the big ideas. Those are the big ideas. So I'm going to stop recording now. I've got a couple people online. I'm going to go ahead and answer their questions. But I wanted to share with you these big ideas, what's going to be on the Chapter 5 and Chapter 7 test. Thank you so much for watching.